Hello there, my name's Neil Spooner. I'm the chair of the Patient-Centric Sampling Interest Group, and we'd love to welcome you to our latest webinar, Recent Workflow Advances and Novel Applications in Volumetric Microsampling Supporting Quantitative Bioanalysis, brought to you by Jeff Plomley and Ming Chen from Altasciences. And before we hand over to Jeff and Ming, I'd just like to take a moment to thank the sponsors of PCSIG who make it possible to bring these webinars to you. Very much looking forward to this content. If you would like to submit any questions during the presentations, please use the chat function on your browser, and we'll come to those at the end of the presentation. So with no further ado, I'd like to just note we are recording this whole webinar, and it will be available on YouTube a week or so after the event. With no further ado, I'd like to hand over to Jeff. Thanks, Jeff, and I'll go off stage.
Okay, well, thank you, Neil, for the introduction, and we're certainly happy that we're able to contribute to this wonderful patient-centric sampling interest group series of really outstanding webinars. So today we're going to talk about some of the exciting projects and recent advancements at Altasciences in the microsampling space over the past year. And to set the stage, I'll start off by highlighting the most common microsampling devices that we've supported over the past decade or so. And then we'll dive into three primary bioanalytical challenges associated with dried blood microsamples, and an experimental strategy that aids in actual device selection. We'll then go into an introduction to impact-assisted extraction as a sample preparation workflow to mitigate some of the attributes of rigid porous polymers. And then we're going to look at some microsampling case studies that exemplify the relevance of the sampling substrate. And then look at two case studies, one for anti-tuberculosis drug panel and antisense oligonucleotide applications, both of which are extremely relevant as therapeutic applications. So I'll start with the microsampling device overview. And all of these modern devices on the screen here, they all offer volumetric blood collection without hematocrit bias. Now, I've divided the volumetric microsampling devices based upon the substrate material. As our experience has taught over the years, that this can have a major influence on analyte recovery, stability, and the implemented extraction workflow. So these are the three most commonly supported devices in our laboratory. So we have the Mitra and the Tasso as rigid porous polymer substrates for finger stick and vacuum assisted microneedle on-body collection respectively. And there's been recent design improvements in the revised version of the Tasso M20, which they now call the T20, that has additional venting in the tile to reduce drying time by 30%. And that's important as we've had applications at Altasciences in the M20 wherein analyte degradation was noted due to prolonged drying periods in a wetted state in the presence of hemolyzed blood. Depending on your analyte, that can be extremely deleterious. There's also higher capacity overflow pads that absorb more blood and therefore reduces overfill. The Lancet has also undergone a design change. Rather than a 3.5 millimeter incision depth, it's now 1.6 millimeter. So expectedly, that's going to reduce sampling pain even further. The surfactant coating on the tile has changed. It doesn't hydrolyze on the shelf. And so that's expected to improve stability of the fluid flow. And in clinical testing, Tasso has reported that the T20 filling of all 4 samples was 13% more successful than the M20. On the cellulose side, and the cellulose used in the Capitainer B product is an Alstrom Gray 222, so it's an untreated cellulose. It comes in a rather friendly foldable card format that leverages a discrete collection inlet for fingerstick blood that then fills an exact volume integrated capillary for deposition onto that cellulose substrate. And I'm going to go into more details of the Capitainer shortly, since the anti-tuberculosis and the antisense oligonucleotide case studies leverage this device. But it is worth mentioning that the rather ingenious card format is readily amenable to other engineering designs, as indicated in this slide. So it really has been a universal platform for engineering microsampling advancements. And here you can see the rate of product development and the scope of matrix types that are covered. You know, since the talk is recent advancements in microsampling, I thought that these were worth mentioning. So since the introduction of the Capitainer B, which was previously called the QDBS back in 2020, the B50 was launched 2 years later, and that allows for greater blood volume collection, which can aid in achieving lower detection limits where necessary. The SEP10 is a more recent introduction, and that contains a hematocrit removal membrane in order to generate dried plasma spots. And that can potentially solve problematic assays where hematocrit is a root cause challenge. And it also facilitates workflows focused on plasma-based assays while still maintaining the volumetric accuracy for sample collection. It may simplify bridging studies as well, although the extent to which dried plasma is equivalent to centrifugal plasma really remains hazy, and there's more work that's required in that space. And finally, a card dedicated to dried urine significantly simplifies remote collection for this matrix. And I'll just mention that there's a terrific article recently published in Advances in Sample Preparation that nicely details applications from this suite of cards, including the B-Vanadate card, which I haven't put up here. Since it's very application specific, it's really for phosphatidyl ethanol, PEth. And within that card, there is a treatment with a phospholipase D inhibitor. So Capitainer has deep expertise in developing these strategies to integrate analyte protectants in their product. And this will come into play shortly when we talk about the anti-tuberculosis drug panel as a case study. And finally, one of the more recent advancements of the card platform is the ability to accommodate integrated electronics powered by metal-free bioenzymatic fuel cells and active sensor tags that have been developed by BEFC. And what that allows is the capture of sampling time whilst also recording temperature and humidity exposure during sample shipping. And this really is a tremendous breakthrough that significantly decomplexifies these critical logistics that are associated particularly with remote sample collection. And this has come about as a result of a collaborative effort between Capitainer, BEFC, and VTT, which provides the roll-to-roll research expertise that's focused on point-of-care diagnostic systems. And all this is funded by the EU Project for Sustainable Functional Electronics. In terms of how the device works, the clock passively activates when matrix enters the system, and this is for the SEP10 as exemplary, but the technology works on all of the card formats for the collection of all the matrices, except I believe for the urine card. So because it's passively activated, there's no user intervention. The resolution of capture time is 1 to 2 minutes, and the data is currently read out via NFC to a smartphone, which is exemplified here as a demo, but in the future, it'll be read out through RFID, so radio frequency identification, and that'll go directly into a LIMS system. Okay, so even though the three most predominant devices that we support in our lab all offer volumetric blood collection regardless of the hematocrit level, there are other interrelated challenges for the downstream bioanalysis. And these include hematocrit-induced recovery bias, where extracted analyte yields decrease with increasing hematocrit. And that was first noted by Mano et al. Back in 2015. And this tends to be more predominant with the highly porous rigid polymers, the tasso, the mitra, and that's due to increasing erythrocyte concentrations that can trap analytes in the pores of the media. And in bioanalysis, it's important to recognize that because our internal standard is not on the sampling substrate, it cannot compensate for variability in analyte recovery. In addition to decreasing recovery with increasing hematocrit, we've also correlated increased blood hematocrit with analyte instability for some molecules. And we'll have a case study to highlight that shortly. But moreover, the trapping of moisture in these porous polymers can lead to drug transformations resulting from hydrolysis, oxidation, and other enzymatic-driven processes in the presence of lysed RBCs. And of course, this is analyte dependent. To further convolute matters, an apparent analyte instability may be age-related extractability. That is, as the microsample ages, reduced analyte response is due to a loss in recovery and not due to instability, in which case an alternative sample preparation workflow might be in order. And this was first identified by Chi et al. At the Merck lab, where an initial direct extraction approach led to a 20% reduction in response for microsamples that had aged 7 days when compared to 24-hour storage. And then when they went to a more aggressive sample preparation approach, involving vortex mixing, sonication, and liquid-liquid extraction, they achieved consistently high recovery, regardless of the age of the microsamples, up to 35 days titrated. And finally, we have observed that as hematocrit increases, age-related extractability can become more pronounced. And therefore, the age of the microsamples and the hematocrit level one optimizes extraction methodology on can have an enormous impact on assay success. In our experience, workflows that eliminate hematocrit recovery bias have also successfully eliminated age-related extractability. And we'll discuss that workflow shortly. And again, a lot of these issues are associated with rigid porous polymers. So in light of the discussed bioanalytical challenges, we need to consider a strategic approach to method development that not only allows for the elucidation of those mechanisms that can influence assay performance, but that will also aid in device selection. That is, an experimental strategy that allows one to tease out which potential mechanism might be at play when observing analyte response. Is it stability or changes in dried blood morphology that result in age-related extractability? In other words, high recovery initially that decreases over time, or is it hematocrit-induced recovery bias due to trapped analyte? So this is really a bioanalytically driven approach in the absence of ClinOps preference. Sometimes sponsors will come to us and unequivocally they have already decided on what device to use, and that may be driven by the patient population. Other times they have no idea what device to use, and there we rely on the bioanalytical attributes of each device to help drive that selection process. So how does this experimental strategy work? Well, we optimize our recovery experiments based on microsamples that are prepared in the following fashion. We use low and high QC concentration levels to determine linearity in response at hematocrit levels of 25, 40, and 55%. So there's the hematocrit influence. And that's applied to both rigid porous polymers. So that would be tasso and mitra, as well as cellulose, which in our case is the Capitainer B product. We then store those microsamples at -20, 4, and room temperature, and under conditions of accelerated stability testing at 40 degrees and 90% relative humidity. We then define a reasonable storage time over which to titrate our recovery assessment as a function of hematocrit, substrate, and storage temperature, typically up to 28 days, which falls within the range of method development time for completion. Now, if the reduced temperature QCs have a higher response than those freshly prepared and dried 24 hours at room temperature, then we know right away we have an initial degradation of analyte upon drying. In that case, we will store our freshly prepared QCs at reduced temperature for 24 hours or more for an accurate assessment. So with this experimental design, we can examine the interrelationship between hematocrit level, analyte stability, and age-related extractability, while also determining the role that the sampling substrate might play in either exacerbating or mitigating each of these occurrences. And we've leveraged this experimental strategy on a large number of programs with great success. Now, in terms of sample preparation to determine the recovery for each of the conditions outlined in that experimental strategy, our experience has taught that a technique which eliminates hematocrit recovery bias, again, also eliminates age-related extractability. And we know from published work that the more aggressive you are in the sample preparation approach for these rigid porous polymers, the higher the success of eliminating hematocrit bias on recovery and age-related extractability. So we designed a workflow that we call impact-assisted extraction. And for the past decade, this has been used on a multitude of assays that have been successfully validated, notably largely without ISR failure. And here's how the impact-assisted extraction works. So as illustrated on the left, into a 96 well plate, we first place our rigid porous polymer microsample, followed by a stainless steel bead. And then we add an extraction solvent, which contains our stable label internal standard. We perform bead beading currently on a Geno grinder model 2010, and through titration, we found that 1750 strokes per minute for 5 to 10 minutes is optimal in the vast majority of cases. So why impact-assisted extraction as a workflow? Well, these rigid porous polymers, although they are rigid, they are compressible. And the goal here is to compress the substrate. And when we compress the substrate, we have expulsion of solvent, which contains dissolved analyte. And then when the substrate is not being compressed, we have reformation of that substrate. And so we get reabsorption of solvent into the RPP. So it's very much like wringing out a sponge, soaking and wringing out a sponge. And we originally published work on this process in Bioanalysis back in 2017. The article is listed here for reference. And in that, we use naproxen as a probe to demonstrate that when compared against a sonication technique that had been optimized, when we compare recovery versus hematocrit up to 63%, from 0 to 63%, 0 being plasma, we find that we get a consistently high response, completely independent of hematocrit. And that has been the case for every single assay we have developed with this workflow from rigid porous polymers, including, again, Tasso and Mitra. Okay, so I just want to briefly show this slide to talk about the relevance of the sampling substrate, because cellulose is easier to handle. You don't need to be as aggressive with cellulose to mitigate hematocrit bias. If there's analyte degradation as a result of matrix, cellulose is not going to help you there. But if you have analyte degradation as a result of moisture trapped in these highly porous regions where all this reactivity can occur during the drying process, then cellulose can be advantageous. So this case study really demonstrates the relevance of the sampling substrate. So the initial work was conducted on the Mitra for this molecule, which I can't show. This is a sponsor's molecule. And so with the Mitra under optimized impact-assisted extraction conditions, we had an average negative 20% concentration bias for stability QCs that had been stored between 7 and 28 days at room temperature in the presence of desiccant, regardless of the hematocrit level. However, when we reduce the temperature storage, we can meet acceptance criteria, so we can rule out age-related extractability. And I'll just mention that the freshly prepared metra calibrants used to determine the stability QC biases were stored at 4 degrees for 24 hours. Recoveries were about 85%, and again, independent of hematocrit. So the totality of the data suggests a kinetically driven process here for degradation, given the temperature-dependent response loss. We did establish 8 hours of stability in whole blood and 5 hours of stability in hemolyzed blood. And I'll just note that hemolyzed blood stability can be a useful diagnostic, since RBCs do rupture upon microsampling drying, and analyte is in a semi-wetted state in these rigid porous polymers, and that can play a role in drug transformation. So given the liquid blood stability results, one must question the sampling substrate. And so we repeated our 28-day experiment at room temperature and 4 degrees on untreated DMPK-C cellulose substrate. Now, at the time that these experiments were conducted, the Capitainer B was not on the market. And I'm just simplifying the results table here with data from high concentration stability QCs. And as we can see, stability was achieved on the DMPK-C card substrate at both room temperature and 4 degrees. Now, the fact that we have stability on cellulose substrate allows us to conclude that the drug is chemically stable in dried blood matrix. So our room temperature metra results are not matrix dependent, but rather substrate specific. Further evidence for this can be seen if we sample the analyte in methanol, plasma, and blood, in that we observe less bias with increased shielding of the analyte from the substrate. So these results really were the impetus to include cellulose as part of our screening strategy when selecting devices. Okay, next I just want to delve a little bit more into the Capitainer B device, since this will set the stage for the anti-tuberculosis drug and ASO applications to follow. And it will aid in visualizing how some of the experiments were conducted. So its architecture incorporates an extremely accurate and precise microfluidic channel for the volumetric deposition of 10 microliters of blood, as previously mentioned, onto Alstrom 222 discs, which are pre-cut, and they have a capacity of 20 microliters total. 20 microliters is an important number. It has a discrete sampling inlet, so it is not a direct to substrate collection such as the metra. And it's cleverly engineered with a waste absorbent that collects blood volumes that exceed the capacity of the capillary channel. And in this manner, one simply cannot oversample on this device. So we sample a blood drop from a finger stick, and then wait around 20 seconds or so for the blood spot to appear in the translucent window. I just realized that I have a pointer. Okay, that's going to be very helpful. So here's the sampling inlet. Here's the translucent window where we can see that we have successfully collected our blood spot. And then the microsamples are easily removed from the back side of the card. These pictures here were the original Capitainer Q DBS. So this is the updated Capitainer B packaging. But it's very easy to open these flaps on the back of the card and pluck off the dried blood cellulose substrate. And so tampering is very easily detected. If you get the sample into the lab and these have been opened, then you can suspect that that sample has been potentially tampered with. Now, although the capacity of the substrate is 20 microliters, the blood volume collected is 10 microliters, and you get 2 spots per card. Sorry, I skipped ahead a little bit. So mechanistically, how does the device work? Well, it controls the timing sequence of events by the thickness-dependent dissolving time of that thin dissolvable film, or TDF. So when a droplet of unknown blood volume is applied to the inlet. The capillary microchannel fills, as shown here, and that fills to capacity. And then the TDF at the inlet starts to dissolve. And once the inlet TDF dissolves, there's a breakthrough of blood onto this waste absorbent here. And so all of the excess blood that does not fill that capillary channel goes to waste. And that's why we cannot oversample. And then once we have this pinch off here at the inlet capillary microchannel, the TDF at the outlet then starts to dissolve and the microchannel volume empties through the outlet. And that's facilitated by the capillary forces that occur during absorption of blood onto the cellulose disc. Okay, so let's move into one of the key applications that has recently been developed in collaboration with Gates MRI as our sponsor for this program. And this was done in 2025. So this is quite recent. And it exemplifies a number of the principles and strategies that I've laid out. So the assay objectives were to develop a quantitative dried blood assay for strain-resistant tuberculosis drug panel regimens, leveraging the Capitainer B microsampling technology for a therapeutic range of 10 to 10,000 nanograms per mil. And there were several challenges in this assay. One is we needed to make it straightforward and simple to implement in terms of the sample preparation approach, because this assay needed to be transferred to multiple labs where the tuberculosis studies were going to be conducted. And so one of the questions, of course, is can suitably high and linear yields from cellulose be achieved for all of the analytes in the panel? And will recovery biases due to blood hematocrit be mitigated with a less aggressive extraction approach? Homogenization is not an option here. We're dealing with an unprecedented drug combination in the panel with a very large polarity range, and literature reports indicated limited stability for several analytes. And so the anti-TB drug panel consisted of 5 drugs, 2 metabolites, cetelazide sulfoxide, cetelazide, quabodepistat, and pretomanid. And then the problematic analytes, delamanid, endesmethylbedaquiline, and bedaquiline. And in terms of log P, calculated log Ps, we're going from a negative 0.1 to 7.2. So a pretty broad polarity range. In terms of optimized extraction, it looks like this. And one of the nice things about cellulose as a polyhydroxy compound is methanol is highly effective as a hydrogen bond disruptor, as a pre-soak, for example, leveraged with an orthogonal approach with a aprotic solvent such as acetonitrile. And so that's exactly the approach we took here. So we agitate for 30 minutes in a vortex mixer the disc alongside a stable label internal standard mixture in 50-45 5 acetonitrile methanol water. And then we add extraction solvent and sonicate, centrifuge, aliquot off the supernatant and dilute with mobile phase, dilute with a mobile phase compatible conditions to run this assay under gradient conditions that allows for suitable separation of each of the drugs in the panel, which I've shown here. Fortunately, they all ionize by positive electrospray, so we did not need to do any polarity cycling. The detection limit was such that we had a reasonable extract dilution factor to mitigate any sorts of matrix effect. And successful chromatography was achieved with a polar RP, which is just an absolutely fantastic phase for retaining really high polar compounds with a suitable K prime. And then, of course, the gradient was such that we could keep the run times to reasonable periods of 5 minutes for the full panel. So BDQ and NDB, that is bedaquiline and its endesmethyl metabolite, are known to have limited room temperature stability in dried blood, as recently reported in Journal of Chromatography B. There's not much more than 10 days stability at room temperature for these two analytes. Now, if you reduce the temperature, you have increased stability of 313 days for bedaquiline, 53 days at 4 degrees for its metabolite, and 313 days at -20 and -80. But what's the point of doing microsampling if you then just have to store the samples at reduced temperature? So what was interesting in this report is that the degradation products were not identified, and there was no attempt made to stabilize the degradation process other than the reduced temperature. So we set out to investigate that and first looked at the relationship between the storage temperature and the blood hematocrit, something not previously reported. And after 6 days of desiccant storage at room temperature, 4 degrees and -80 at 25, 40, and 55% hematocrit, we could corroborate the temperature dependency on stability, but moreover, we could observe decreased stability with increased hematocrit regardless of the storage temperature. And I've shown that here. So we see increased stability with reduced temperature, but regardless of what temperature you're at, there is a consistent trend of decreased stability with increased hematocrit. So when we observe analyte instability in dried blood, one thing we look at is forced degradation studies of API, since in our experience, this can identify potential high-risk transformations. And in the case of bedaquiline, an oxidized product was identified as the major degradant. So we therefore screened for this oxidation product under targeted MRM conditions for our aged microsamples at 55% hematocrit under each of the storage conditions, and could identify a clear trend of increasing expression level with increased storage temperature. So this was most certainly the process that was occurring on these dried blood samples. Turning to the other problematic assay, problematic analyte, delamanid. It's reported to be metabolized to the 2-amino 4,5-dihydroxazole derivative by albumin in plasma. However, its formation can be arrested at pHs equal to or less than 6. So with this collective data, then, we examined a range of potential antioxidants to stabilize these 3 reactive analytes. And in addition to the standard phenolic antioxidant butylated hydroxytoluene, BHT, we also looked at chelating agents, because we know that when we see increasing hematocrit results like that, it fits with the known role of iron ions as oxidizers. And the fact that the liberation of iron from hemoglobin during air drying enables the production of hydroxy radicals through a Fenton reaction, it made sense to arrest the activity of this liberated iron. So in this table, I'm only reporting the optimal results from each protectant, which was added directly and quantitatively, that is 20 microliters, to the cellulose disc of an intact card, which was then dried for 24 hours prior to then depositing 10 microliters of blood at 55% hematocrit directly to the substrate. We then looked at recovery after 6 days of room temperature desiccant storage. And as we can see in this table here, 1 molar of ascorbic acid was the optimal stabilizer for each of the 3 reactive analytes. I've also indicated here the untreated cellulose substrate recovery losses, and we see some 30 to 40% recovery loss for NDB-BDQ, and almost 50% recovery loss for delamanid. When we treat the card with 1 molar ascorbic acid, we are seeing near quantitative recovery. So pre-treating the cellulose in the Capitainer B cards with 20 microliters of 1 molar ascorbic acid, drying for 24 hours, and then depositing 10 microliters of calabrins and QCs direct to substrate, we wanted to see how the assay would perform. And so the next few slides are simply the results of some of the method development stressing evaluations that we conducted. And first up is the between run precision and accuracy. derived from 3 batches with QCs at 4 concentration levels and replicates of 6 for each batch. And as we can see here, all acceptance was met for both precision and accuracy. So that's a great start. The next critical experiments examine the matrix effect from 6 different whole blood donors, whose mean is reported here for each drug in the panel, again, meeting acceptance criteria. We do note a distinct lack of hematocrit effect up to 55%, and that pacifies our fears of recovery bias when using a milder approach to sample preparation for cellulose compared to what we use for rigid porous polymers. And finally, the extraction yields are such that they readily support achieving the LOQ of 10 nanograms per mil using the SciEx 6500 plus with an extract dilution factor sufficient enough to negate matrix effect. So we're pretty much over 70% recovery here. Perhaps the most significant evaluation is the long-term microsample stability assessed at room temperature and as a function of blood hematocrit. And here we've achieved excellent stability for all 3 of our reactive analytes up to 68 days measured. Only cetelizide is borderline, and that might require some further investigation. It's fine up to 21 days. Something happens when we hit the 68-day mark. So sample integrity has been confirmed with that pre-treatment approach. So satisfied with the assay performance using stabilizer, we were then interested if Capitainer could produce cards with pre-treated cellulose incorporated in the final assembly, since our backdoor treatment of the card fouls the device functionality due to dissolution of the TDF that meters the blood collection to the substrate. And fortunately, Capitainer was very supportive of this endeavor and provided enough cards for a test batch to evaluate a within-run precision and accuracy batch, wherein we could compare inlet sampling versus direct-to-substrate in order to confirm that the Capitainer's ascorbic acid treatment approach has no impact on device performance. And then, of course, evaluate the stabilities for BDQ, NDB, and DLM. So we're going from laboratory proof of concept to a product test batch. So here I've tabulated the precision and accuracy. The precision is in brackets of the mean from 6 replicates of low and high QCs applied direct to substrate. So DTS is direct to substrate. And I did that on Altasciences treated cards and Capitainer's card treatments, and also to the inlet for the Capitainer treatment. So we added 25 microliters of sample at the inlet. And the results were back calculated from calibrants applied direct to substrate on the Altasciences treatment. Again, the Altasciences treatment is 20 microliters of 1 molar ascorbic acid onto the substrate, dried 24 hours, and then sample applied to the disc. So there's no significant difference between inlet sampling and direct to substrate on the Capitainer treated cards. And that indicates that the card functionality has not been compromised in the Capitainer treatment. And for the most part, the Capitainer treated cards align with results from the Altasciences treated cards. Finally, the microsample stability on the Capitainer test batch at 40% and 55% hematocrit with room temperature desiccant storage for 14 days indicates acceptable for performance. Delamanid is not quite meeting the acceptance criteria at high QC levels, so perhaps a bit of further refinement might be necessary. But delamanid really is the most reactive species on untreated cards. We observe 50% degradation within 24 hours in the presence of desiccant. But overall, fantastic results. I'm going to skip the key highlights. I want to leave enough time for Ming because he has some outstanding ASO data. But as Neil said, these can be revisited at any time. Just one thing to point out, though, is that this was the first application of volumetric microsampling applied to this particular unique anti-TB drug panel. And then I'd just like to, before I hand this over to Ming, I'd like to thank the contributors for this research on this slide, notably Deemika Rossi at Altasciences and our sponsor for the anti-TB work, Anaksha Wikram Singh from Gates MRI, and all the folks at Capitainer, which have been extremely supportive over the years, and to Par, Olis and Lido, and his team for their collaboration in working out a strategy to incorporate pre-treated substrate without fouling the card functionality. Okay, Ming, over to you.
All right. Thank you, Jeff. Hi, everyone again, and thank you again for joining this webinar. I will be presenting our work on the microsampling application for antisense oligonucleotides, focusing on two microsampling platforms, Mitchell and the Capitainer B, coupled with hybridization LC-MS approach. In this presentation, I will start with a brief introduction to antisense oligonucleotides and the key bioanalytical challenges specific to determining the ASO from the dry blood microsamples. I will then present two case studies using the Formiversion as a model analyze, highlighting the workflows and the mass performance from both Mitchell and the Capitainer B platforms. Antisense oligonucleotides are synthesized single-stranded nucleic acid, typically consisting of 12 to 25 bases. They are designed to bind complementary RNA sequences to modulate gene expression by inhibiting translation or altering mRNA splicing, leading to protein level changes. Quantitation of the ASOs from dry blood microsamples presents several unique analytical challenges. First, standardized small molecular microsample extraction approaches cannot be directly translated, as the desorption solutions containing organic solvent can cause ASO precipitation, leading to low and the variable recovery. In addition, the high percentage of organic solvent are not compatible with downstream ASO extraction, which typically requires aqueous environment. Achieving sufficient assay specificity is not a major challenge, as the high ASO recovery is often required the use of surfactants that extensively dissolves matrix components from the substrate, increasing the extract complexity. The lower sample volume and the limited ability to concentrate the extract can further compromise sensitivity and negatively impact the achievable lower limit of quantitation. Finally, the presence of the truncated metabolites also require high selectivity to avoid isobaric interference and compromise quantitation. To address these challenges, we employed hybridization LC-MS workflow for ASO bioanalysis, as illustrated here. Following digestion, the biological matrices such as plasma, serum, CSF, or tissue homogenate are incubated with the biotinylated complementary probe pre-conjugated on the strictavidin magnetic beads. After washing to remove the matrix component, the captured ASO analytes are thermally eluted and analyzed by LC-MS, which offers high sensitivity and selectivity with typical LOQ in the range of 0.5 to 1 nanogram per ml from 100 microliters of plasma. In the following slides, I will present two case studies demonstrating how this hybridization LC-MS workflow can be adapted for microsampling. Let's move to the first case study focusing on the Mitchell device. The model analyzed selected was Formiversion, a 21-mer furanate antisense oligonucleotide, which is the first ASO drug approved by the FDA in 1998. Extraction from the Mitchell substrate using impacted system extraction. We screen different extraction solutions to optimize the ASO recovery. For the hybridization-based extraction, the full-length complementary probe was used to ensure the assay selectivity. Together, impacted system extraction and the hybridization enabled efficient, consistent, and producible ASO recovery. Mass quantification assessments, including the sensitivity, linearity, precision accuracy, matrix effect, hemochromatoid impact, and the microsample stabilities. The slides summarize the LC-MS optimization to achieve the robust selectivity for Formiversion and its related species. On the left, the LC-MS conditions are listed, including the iron pairing mobile phase conditions. On the right, the representative chromatogram shows clear baseline separation between formula version, the truncated N-2 and N-1 metabolites, as well as the N+1 and N+2 analogs. This level of chromatographic resolution is critical to accurate quantitation because these metabolites differ by only one single nucleotide and could layer-wise cause isobaric interference with the parent formula version. Effective impact-assisted extraction is essential for recovering aerosols from the mutual substrate. In this study, we evaluate several desorption solutions, including trees and EDTA-based buffer with different salt concentration and the different types of surfactants, such as NP40 and 2020, and the water as the control. The result shows that the solution 1, which contains trees, EDTA, sodium chloride, and NP40, consistently provide the highest and the most linear recovery across the low and high QC samples. Importantly, NP40 was the key component that ensured the quantitative and the consistent recovery, while it is compatible with the downstream digestion and the hybridization steps. For this reason, the solution 1 was selected as the optimal extraction solution. Here we show how extraction from the mutual microsamples is integrated into the hybridization LC-MS workflow. First, 10 microliter mutual dry blood microsamples is placed in a 96 well plate. The N+2 analog of formula version is ended as an internal standard to check the variabilities during the microsample extraction and the hybridization. The optimized aqueous desorption solution containing NP40 is ended along with the stainless steel beads. The impact-assisted extraction is then performed at 1750 strokes per minute for 5 minutes, repeated for 2 cycles. This ensures that the formula version is fully dissolved from the mutual substrate. After the IAE, only the liquid containing the ASO is transferred to the next steps. The mutual tips and the stainless steel beads remain in the original plate, preventing any interference with the downstream digestion and the hybridization, especially during the automatic magnetic washing and the elution. During the hybridization, the extract ASO are captured using a full-length complementary probe washed to remove the matrix components and then eluted for LC-MS analysis. This integrated workflow ensures the quantitative recovery of both formula version and the internal standard across low and high QC level, while mitigating potential hemocratic and age-related extraction bias. This representative chromatogram shows the method performance, including the extracted blank, the LOQ at 5 nanogram per ml, the ULOQ at 1,000 nanogram per ml, and in the internal standard. The peaks display consistent retention times and the peak shapes across the calibration range with a clean background in the extract blank samples. The extract LOQ corresponds to 1.6 picogram of formula version loaded on the column with a single to lowest ratio of 50:1. Next, let's look at the mass linearity and the precision accuracy results. Here we can see the excellent linearity was achieved. Importantly, the low concentration standards fall well on the calibration curve, showing that long specific binding is negligible, which is the most common issue in the oligonucleotide bioanalysis. Precision accuracy also met acceptance criteria across all QC levels based on 6 analytical rounds with 24 replicates per level. Next, we evaluate the matrix effect and hemocratic impact. Matrix effect was assessed at a low and high QC level and shows minimum interference, confirming the selectivity of hybridization-based approach, despite full co-extraction of the matrix components when using the surfactant-containing desorption solution. Hemochronic impact was tested using the QC samples codified against the calibration curve prepared in a blood at the 44% hemochronic level. Low significant bias was observed, showing that the impact-resistant extraction procedure with the optimized surfactant-containing desorption solution provide quantitative and consistent recovery. Microsample stability was also evaluated over 115 days at the room temperature with desiccant. Stability QCs remaining within 6.3% deviation of the 24 hours controls. This result indicates low age-related extractability even after extended storage. In summary, for this case study, we successfully extracted and quantified formiversion from mutual dry blood microsamples. The hybridization LC-MS workflow allowed highly selective ASO capture even in complex matrices and achieved the quantitative recovery with LOQ of 5 nanogram per ml in just 10 microliters of dry blood microsample. Importantly, assay performance was not affected by the hemochronic variation across the wide range from 0 to 66%. Comprehensive mass quantification confirmed the precision, accuracy, specificity, and the recovery. This work established a solid foundation for applying the microsampling coupled with the hybridization LC-MS to ASO therapeutics. And these results were published in Bioanalysis in 2023. Building on the mutual work, we next determined the ASO extractability and the overall assay performance from the Capitainer B cellulose substrates. The hybridization LC-MS workflow was adapted from the mutual assay and further optimized for the cellulose microsamples. The LC-MS configuration was fine-tuned to improve the LOQ while maintaining a robust separation between the parent ASO and the metabolites and the analogs. A comprehensive mass quantification was performed to confirm the workflow is suitable for mass validation and the sample analysis. We also assessed the device immunity to oversampling, ensure the accurate volumetric connection. To begin, we optimized the LC-MS configuration. Typically, low LC flow rates could help improve MS sensitivity in lactic ion mode for ASO. However, low flow rates such as 0.3 ml per minute amplify the pre-colon volume effects and the reduced LC separation resolution. To address this, a post-injection divert valve was implemented. Immediately after the injection, the autosampler is bypassed, allowing the LC pumps to connect directly to the analytical column. This minimizes the autosampler and the tubing depth volume, improves gradient responsiveness, and reduce the peak dispersion. On the right, representative chromatograms at 0.3 ml per minute are sought with and without the divert valve. When the valve is applied, peaks are sustainably sharper and better resolved. The increased peak height directly translates into the improved single-to-noise ratio, which is essential for achieving a lower LOQ. Under these conditions, formiversion and his truncated metabolites and the related analogs remain well separated, enable both high sensitivity and robust selectivity. Following the LC-MS configuration optimization, we evaluated whether impact-assisted extraction using the stainless steel beads was suitable for the cellulose-based Capitainer B device. We can see here, after extraction, the cellulose sticks was completely fragmented. While steel beads are effective for the ASO desorption from mutual, they are too aggressive for the cellulose substrate. As a result, the Mitchell procedure does not translate directly to the Capitainer B and introduce several issues. Fragmented cellulose makes a reliable extract transfer difficult, which can affect downstream hybridization and increase assay reliability, variability. In addition, the cellulose debris can be co-transferred with the extract, interfering with the hybridization capsule and the magnetic base extraction. Together, this impact leads to a variable aerosol recovery and compromise the quantitation. To address this disk fragmentation observed with the bead speeding extraction procedure, we adopt a gentle desorption approach that preserves the cellulose disk. Two strategies were tested: prolonged low-speed incubation at 800 RPM for up to 18 hours without steel beads, and the short high-speed agitation at 1750 RPM for 1 minute. Repeat once or 3 times without beads. Low speed incubation around is not sufficient, particularly at a high hemocritic level, resulting in reduced recovery. Short high speed agitation effectively release the aerosol while keeping the disk intact. One cycle is not enough at high hemocritic levels, especially for the low QC samples, but 3 cycles stimulate hemocritic dependent variability, achieving consistent recovery across all conditions. And this optimized the desorption procedure allows a complete extract transfer without co-transferring cellulose debris, enabling effective hybridization and reliable quantitation. Here we show the representative chromatograms, including extract blank, the LOQ at 0.5 nanogram per ml, and the ULOQ at 500 nanogram per ml, and the internal standard. The blank sample shows a clean background with no interference peak, and the peak for universal and the internal standard are sharp and well defined. The LOQ corresponds to a 670 femtogram loaded on a column with a single to noise ratio 25:1. Importantly, these results were obtained from just 10 microliters of Capitainer B microsamples, which is a 10-fold reduction compared to a conventional plasma-based method that typically require 100 microliters. Next, we look at the method linearity and the precision accuracy results. As you can see, excellent linearity was achieved across the calibration range with low concentration standard fitting well on the curve, confirming the negotiable long-specific binding even at the LOQ at 0.5 nanogram per ml. Precision accuracy are well within the acceptance criteria across all QC levels, with 7 analytical runs and 28 QC replicate per level. Next, we evaluate the matrix effect and the impact of hemocritic. Matrix effect were evaluated at the low and high QC level across 6 blood knots, confirming a high mass selectivity. Hemocritic impact shows no significant bias, demonstrate that the developed agitation method preserves disk integrity while providing consistent and quantitative recovery across hemocritic levels. Microsample stability was confirmed at the room temperature and also under the accelerated conditions at 30 degrees Celsius with excellent stability observed. We also evaluate the inlet sampling volume to assess the Capitainer B devices immunity to oversampling. Across both low and high QC level, consistent and reliable results were obtained with minimum reliability and bias. This result confirmed that assay is robust to the sample volume variation, and that oversampling does not significantly affect the aerosol quantitation. Yeah, building on our experience with Mitchell, we successfully applied to the Capitainer B device for the microsampling based aerosol quantitation using a hybridization LC-MS workflow adapted for a 10 microliter cellulose based microsamples. Formulation was quantified with LOQ of 0.5 nanogram per ml. high-speed conjugation without steel beads preserve the integrity of the cellulose disc, enable complete sample transfer while avoiding interference from the cellulose debris during the hybridization capture and the magnetic extraction. The method performance was robust across the wide hemochromatoid range and demonstrate immunity to oversampling, with a full mass quantification confirming the excellent precision, accuracy, specificity, and the microsample stability. Overall, these results demonstrate that Capitainer B is the reliable platform for low-volume aerosol quantitation. All right, so in conclusion, our work demonstrates that the microsampling combined with the hybridization LC-MS enables sensitive, selective, and robust quantitation of antisense oligonucleotide. Both Mitra and the Capitainer B device perform well using only 10 microliter dry blood microsamples, a substantial reduction compared to a conventional plasma-based assay. The workflow is highly specific and consistent, even when surfactants are used for the analyzed desorption. And the assay performance was impacted by the hemochromatoid variation or oversampling, confirming the reliability of the method. Overall, this result shows that volumetric microsampling with hybridization LC-MS is a flexible and a robust approach for aerosol bioanalysis across different microsampling platforms. All right, thank you much for your time. Jeff and myself will be happy to answer any question.
Thank you, Ming. Thank you, Jeff. We've kind of run out of time. We've only got time maybe for one question coming from Hua, who asked, what device speed agitation, 1 minute times 3 cycles, would a vortex mixer work?
Yeah, I guess it's related to the oligo application. Yeah, we use the genome grinder, which is the same instrument we use for the impact assistant extraction. We also try to use the vortexing or sonication, and even we up to the 18 hours. But the recovery is not very consistent, and we are not achieving the quantitative recovery with these low-speed mixing procedures.
Thank you, Ming. And I guess one question from me to both of you, these brilliant methods that you've developed here for your different analytes, have they been used for clinical studies for patients at all yet?
Give you mute.
Yeah, hi, Neil. I'm not 100% sure at this point in time what Gates MRI intentions are for this assay. I mean, we were tasked with developing the anti-TB panel. Is that those particular analytes in that panel had never been done before. They were quite challenging. I can understand why they've not been done before. The stabilities were most certainly a challenge. Now, how Gates is going to roll that out and transfer that to other labs where their trials are going on, we're not aware of at this point in time.
Okay, thank you, Jeff. Thank you, Jeff. Thank you, Ming. And thank you to all of our audiences for attending our webinar today. For those of you that are not aware of the PCSIG, there's just a little bit of information about us there. The QR code on the left will take you to our website where there's a lot of information. The QR code on the right will take you to our LinkedIn group. We welcome all members. It's free to join. Please come and contribute to our mission of getting knowledge of patient-centric sampling out there, understanding the challenges, and just to remind you that we've got a series of webinars coming up. We've got one coming up on February the 10th with Jaden Lee Roberts, and we've got another one on March the 17th with Giovanna Forte, very much looking at different aspects of patient-centric sampling. We currently have an agenda of webinars booked up to February 2027, but if you have any suggestions for webinars, please get in contact with us. Before we leave, just like to thank all of our sponsors again that enable us to do our wonderful work, and thank our speakers again and you as the audience. The slides and the presentation will be available on the PCSIG YouTube channel in a week or so, and also through the PCSIG website. Thank you everyone for your attention.